What if a single edit to your DNA could change everything? No therapy, no medication, no years of intervention. Just a rewrite at the molecular level. Scientists now have a tool called CRISPR that can do exactly that. Locate a specific sequence in your DNA and change it. And people are asking, could it work for autism? Here's what the science actually says, and here's what it doesn't. Before we go any further, let's be clear about something important. This channel is built on the belief that neurodiversity has value. Neurodiversity means that differences in how brains work, including autism, are natural human variations, not simply errors to be corrected. This video is not about erasing that. It's about understanding where science is genuinely trying to reduce suffering. Things like life-threatening seizures, loss of motor control, and severe developmental regression, while respecting the full humanity of every autistic person. That framing matters. Hold on to it. I want you to meet a family. Not a real one, but one that represents thousands of real stories. Imagine a little girl. She hits every milestone in her first year. She babbles, laughs, reaches for her parents. Then, somewhere around 18 months, things change. She stops using her hands purposefully. She loses words she'd already learned. Her breathing becomes irregular. Seizures begin. Her parents search desperately for answers and eventually hear 3 words: Rett syndrome. A single gene, MECP2, isn't functioning the way it should. This is the reality that drives researchers into the lab every single day. Not a philosophical debate. A little girl who used to laugh, and a family who would do anything to help her. Keep her in mind as we walk through the science. Now, here's what makes this so difficult. Autism is not one disease, one gene, one mutation. It's a spectrum shaped by hundreds of genes, environmental influences, and early brain development patterns. Trying to edit autism out broadly would be like trying to fix the entire internet by correcting one line of code. It simply doesn't work that way. But within that spectrum, there are rare conditions where a single gene plays a dominant role. And that's where CRISPR enters the conversation. Not as a cure for autism, but as a potential tool for specific genetic disorders that sometimes include autism traits. That distinction is everything. Let's look at 3 of those conditions, starting small and building up. Gene number 1, SCN2A. This gene regulates how neurons send electrical signals. Think of it like the timing mechanism in a circuit board. When it misfires, the whole system is thrown off. Researchers are exploring ways to reactivate the healthy copy of this gene, not to change who someone is, but to reduce specific neurological symptoms. Gene number 2, SHANK3. SHANK3 builds the structural scaffolding of synapses, the connection points between neurons. When it's missing or disrupted, those connections weaken, like a bridge losing its support beams. This leads to Phelan-McDermid syndrome, which often includes autism traits. Gene therapy approaches are now being explored to restore that missing protein function. Still early, but the direction is clear. Gene number 3, MECP2 and Rett syndrome. Remember that little girl? This is her story at the genetic level. For years, scientists assumed Rett syndrome was irreversible, that once those developmental windows closed, nothing could reopen them. Then, in animal models, restoring MECP2 function produced something extraordinary. Breathing stabilized, motor coordination improved, functional visual processing was restored. Rett syndrome is not the same as autism, but this finding sent a shockwave through neuroscience. Some conditions once considered permanent may still be biologically modifiable. That changes everything about how researchers think about the brain. If gene editing could reduce severe disability or life-threatening seizures, without changing your child's personality or identity, would you want it? There's no wrong answer. Drop your thoughts in the comments. I read every single one. Even when scientists identify the exact gene involved, editing the brain is a completely different challenge from editing other parts of the body. Here's why. The blood-brain barrier. Think of this as the most exclusive VIP bouncer in existence. Its entire job is keeping foreign substances out of the brain. It's brilliant at protecting you and brutally effective at blocking treatments from getting in. Scale. The brain contains roughly 86 billion neurons. To put that in perspective, that's comparable to the number of stars in the Milky Way galaxy. A therapy needs to reach a meaningful portion of those cells to make any difference at all. Dosage sensitivity. Many brain genes need to be tuned to exactly the right level. Too little activity causes disease. Too much causes different problems. The margin for error is extraordinarily small. Timing. Neural circuits form early in life. Once established, they're far harder to reshape. The brain cannot be reset like restarting a computer. You might improve signaling, you might reduce seizures, but you cannot rewind development. That's the biological reality every researcher in this field is working within. CRISPR is not theoretical anymore. It has already been used to successfully treat patients with sickle cell disease. Real people, real results. The technology works. But here's the key difference. Blood cells are relatively straightforward to edit. You can remove them from the body, edit them in a lab, and reintroduce them. The brain doesn't work that way, which is exactly why it remains one of the most challenging frontiers in all of medicine. There's a distinction in gene editing that rarely makes the headlines, but it should. Think of it this way. Somatic editing is like fixing a typo in one printed copy of a book. You're treating one individual. The change stops with them. Germline editing is like changing the original printing press. Every book printed from that point forward carries the alteration, including all future generations. Most of the global scientific community strongly opposes germline editing for enhancement purposes. The consensus is firm. Treating severe disease in an individual is one conversation. Altering the genetic inheritance of future generations is an entirely different one. Autism sits right at the intersection of both debates, because for many autistic people, autism isn't simply a medical condition. It's part of how they experience and move through the world. That's why the most responsible researchers are explicit. The goal is not to erase neurodiversity. It's to address severe medical suffering, seizures, regression, loss of function, in cases where a specific genetic condition is causing serious harm. And critically, that conversation must include autistic voices, not just scientists, not just parents, autistic people themselves. The future of autism science won't come from a single breakthrough. It will come from understanding complexity, genetics, development, environment, and the extraordinary adaptability of the human brain. CRISPR is one tool in that journey, a powerful one, but not a magic solution. And certainly not a simple one. The real story was never about editing autism out of existence. It's about understanding the brain deeply enough to help people live healthier, fuller lives on their own terms. If you want science explained clearly, without hype, without fear, and without losing the human story behind the research, subscribe to Bee as a Butterfly. Next week, we're going inside a real-world SHANK3 gene therapy trial. What's actually happening in the lab, what families need to understand, and why the headlines will almost certainly get it wrong. You deserve the full picture. Subscribe so you don't miss it. And if this video made you think, share it with someone who needs to hear it.